Hello, good morning, everyone. My name is Mahesha Manjunatha Pujari, and welcome to the day 3 of the GAN program. The next generation sustainable alternative proteins for food fundamentals and technological innovations. I'm not going to cover every spectroscopic technique, but I'm going to cover some of the most important things that we use today. Okay. I found this figure from this particular website. I really enjoyed it. So basically, when it's a protein characterization, we talk more about the structure. Okay. And then we also talk about the molecular weight, because each protein will have its own molecular weight, and the molecular weight is a fundamental aspect of the protein that again determines the solubility, functionality, and so on, right? And the charge, we discussed about charge also in our very first class: positively charged proteins, negatively charged protein, amphoteric proteins are amphoteric, right? Then we have an isoelectric point, right? And then, as I mentioned in my previous slide, protein if never stays alone in a food system. It will always try to interact with other biomolecule. Other biomolecule could be protein itself. Okay, protein can always interact with another protein. In addition to that, protein can interact with carbohydrates, polyphenols, lipids, and so on. So all this will change at the end of the day, the functional properties of proteins. And we also have something about protein purity and post-translational modification. That means especially when plants generate proteins and they never simply exist as an individual beautiful proteins like an amino acids, as I showed in my previous slide, one of the amino acids may be modified. There may be phosphorylation, there may be glycosylation. So amino acids might be modified to a lesser or greater extent, right, within the cells itself. And that will again change the functional properties of the protein. Please note this point. So amino acids can be modified within the plant matrix itself before you extract and use it. And that is called post-translational modification. For example, phosphorylation is one of the most common modification. You'll add a phosphor moieties to the proteins. Glycosylation, you'll add a sugar moiety to the proteins, and that will change the solubility of the protein. And to identify all this modification, we need to start characterizing the proteins using different technologies. Okay, so I'm going to cover some of these technologies today. I must tell you that I'm not going to cover everything because I don't have that much time, but I'll only show you some of the most important ones we are using today. Now let's move on to the structure. In my very first class, I mentioned to you that proteins are having 4 different structures. All right. Primary structure, it's simply nothing but its amino acid composition. And the secondary structure is nothing but you have hydrogen bonding, proteins to coil, protein act as a sheet. We call it as alpha helix and beta sheets, and also something called random coils. And in addition to that, proteins can be 3-dimensionally folded. Proteins are never linear in food system or in biological system. They are actually 3-dimensionally folded because of, let's say, disulfide bonds, hydrogen bonding, hydrophobic interactions. So the proteins are bent in a way that it gives a particular shape to the protein, right? And that's called tertiary structure. And tertiary structure is also important for protein-protein interaction, protein-carbohydrate interaction, protein-polyphenol interaction, and so on, right? And then we have a quaternary structure where different protein subunits can make a dimer, trimer, hexameric structure of very big complexes of protein. So these are the 4 different possibilities or the structural variations a given protein can have. Now, I'm going to show you a little bit about an overview of how different techniques can be used to reveal these structures. Can you tell me why the primary structure is important? It's an important measurement we have to do, the primary structure analysis, because it also stores the genetic information, because proteins are produced based on the translation within the cells, right? The amino acid composition is also very important because that will talk about the structure and also for example, how disulfide bonds can be formed within the primary structure, and that talks about again stretchability of the matrix, the Dow here, and that's in other words is the functionality. Primary structure is the fundamental, the basis of the proteins. Basically, based on the primary structure, then the secondary structure takes place, and then tertiary, and then the quaternary, because the amino acid composition and the sequence of the amino acid is very important to determine the final tertiary or the quaternary structure of the protein. Fully agree with her. Now we mentioned about 2 points here. One is composition. Composition is nothing but how much lysine we have, how much glycines we have, how much tryptophan we have. But the protein is not just having how many of these amino acids. It's also important to learn how they are oriented, how they are in a sequence, like whether we have a glycine after lysine and tryptophan, or we have a different combination. Okay, I have tryptophan, lysine, and glycine. Now these two, let's say tripeptides, are very different. Do you agree with me? A sequence is also very important, because based on the sequence, the pI of the protein can vary, because the neighboring amino acids will determine how the other amino acids behave. So that's the reason why the sequence is also important. In addition to pI, the sequence also determines how protein can bend and fold. For example, if you want to have a disulfide bond, imagine the disulfide bond has happens because of let's say 2 thiol group. When they join together, you get disulfide bond like this one, right? If they are far away, if the protein cannot bend, the disulfide bond never happen, right? If they're too near, then you still have a very steric strain. They cannot react together. They should be present in a particular distance. The protein should be able to bend. The bending is also dependent on which amino acid is present in the backbone. Make sense? And that's the reason why how individual amino acids are located one after another, the sequence, that's also very important, right? And that determines the secondary, tertiary, and the quaternary structure. So determine the primary structure. The first thing we do the amino acid composition analysis, often done by HPLC or GC. Then we do, to find out the sequence and also the composition, we're going to use Eadman degradation, right? And then we can also use mass spectrometry to determine the sequence, but it's a little bit difficult, especially if you have a very big protein. If the polypeptide of like 20, 30 amino acids, you can easily find out sequence. But if it's a huge proteins with like 3,000 or not 3,000, 300 to 400 amino acids, it becomes a little bit tricky. So often we use Eadman's degradation to find out the protein sequence. I'll get back to that later. Then we have a protein secondary structure. Remember, it is I have only shown the alpha helix here. We also have a beta sheet. And the secondary structure can be determined by FTIR and CD. CD is circular dichroism spectroscopy. And the tertiary structure, we can use fluorescence spectroscopy, X-ray, NMR, and also electron microscopy. Okay. And the quaternary structure, you can use dynamic light scattering, size exclusion chromatography, X-ray, NMR, and so on. Please note that one technique will never give you the full information, right? That's a problem or the difficulty I'm mentioning here in protein analysis. It's often combination of the different techniques gives you the overall structural information. Please note that in order to get a structure of one protein, it might be 3 PhD work. I'm telling you, it's not easy, right? For example, you isolated one particular protein from like a new plant-based matrix, and nobody knows what protein is it, right? Now you want to know what is the nature of this protein. Then you will start with analyzing its primary structure. Okay, I know its amino acid composition. And then you will analyze, okay, its secondary structure. Then you will analyze its tertiary structure. And then you will analyze whether it can make a hexameric structure and so on, quaternary structure. And all these things are very, very tedious processes. Of course, we do have now automated system. We do have artificial intelligence to help us in structure determination. However, it's a very, very complicated and tedious processes. Okay, as I mentioned, I'm not going to cover everything, but I'm covering some of these things. And X-ray is an important tool to measure protein structure. Very, very nice technique. Gives almost complete information about protein composition and also the folding. And I think one of our colleagues will be talking about X-ray stuff in one of the lectures, just to highlight. Okay. Now I'll talk a little bit about the primary structure determination. So you know that protein has amino acids. These are all amino acids. Okay. And there is something called N-terminal and there is something called C-terminal, right? And to determine the amino acid sequence, you need to know which is present one after another. lysine after cysteine and glycine, leucine, isoleucine, and so on. We need to find out which one is present one after other, right? To determine that, what they do is they use a reagent called Eadman's reagent. It's called Eadman's degradation, and this reagent interacts with the N-terminal amino acid, the last one. Okay, and then we get a compound called phenyl thiocarbamyl derivative. It looks like this one. It's a fake structure. And then what we do is that then this reaction is done in alkaline condition, and then we heat it, and we make it a little bit acidic, and then this particular group will be cleaved off here, and then we get an individual unit. Okay, now it's connected to an amino acid. Can you see? This is an amino acid, and we have a derivative, and this can be analyzed by, let's say, HPLC, right? So now I know let's say I don't know what is the S amino acid. I don't remember the proteomics in serine. Okay, now let's say it is serine. Now we came to know that the N terminal, the first amino acid is serine. Okay, then we lost this one. Do you agree? Okay, I can't see it. So we lost this particular part. Then we have again another N terminal here. Do you agree? And again this reagent is attacking here. Again we cleave it. Now you get another structure called something looks like N. Then you analyze it again by HPLC. So like that you will start degrading them one by one, right? And at the end of the day you will realize, okay, this is the final sequence of my protein. Looks really a lot of work, right? Imagine if you have 500 amino acids, you have to do a lot of work. Now it's kind of automated. We can actually do the sequencing analysis. All right, now let's move on to little bit. This is something we use a lot in food science. That's called determination of the secondary structure, because the secondary structure determines the stability of the protein. How stable is your protein, and how stable is your tertiary structure is also dependent on the secondary structure. So we often measure the secondary structure. Why? Because food is often processed. What is mean by processing? We apply light, we apply heat, okay, we applied shear forces, we apply pressure on the food. All this processing is done on the proteins, right? And when you do processing, the protein will change its structure, right? And remember my very first class, the secondary structures are stabilized by something called hydrogen bonding. Do you remember? So the hydrogen bonding is the one that stabilizes the secondary structure. Hydrogen bonding most commonly. That's of course there are some other weak interaction, but hydrogen bonding is one of the interactions that stabilizes the secondary structure. Now we all know that the hydrogen bonding is temperature dependent, right? If you heat it, hydrogen bond breaks. Just to give an example, like let's take water, right? Water contains extensive hydrogen bonding. Do you agree? Water exists in three forms, yes? Ice, solid, liquid, and gas. And the three phases of water varies depending on the extent of hydrogen bonding. In the gaseous, you don't have much hydrogen bonding, so the molecules go apart, so it becomes gas. In liquid, you have a hydrogen bonding, so that brings the molecules together, so it becomes liquid form. In solid, they are tightly packed because of an extensive hydrogen bonding networking, then it becomes ice. So hydrogen bonding determines the phase of the structure, basically. Remember that one. And hydrogen bonding is also heat sensitive. It can be broken. So since you're processing food, there may be the structural changes. There may be the secondary structure change in the protein. And that information can be brought by FTIR spectroscopy. Now, I'll show you how exactly the hydrogen bonding is present in protein. You can see this is a carbonyl group. Can you see here? C double bond O, the carbonyl group. And can you see an NH? Right, this is from another amino acid, the NH, right? And there is a hydrogen bonding between two, right? Electronegative and electropositive group, there is an hydrogen bonding, right? And that pulls the molecules together to make a bending or coiling and so on, right? That's the hydrogen bonding. And now, because of changes, this hydrogen bonding can be broken. When the hydrogen bond broken, now the bent structure can be, the coiling might change, right? Originally it was nicely coiled, now it's maybe weakly coiled, and that indicates the structural change of the protein. To evaluate that one, we are going to use FTIR. IR is a radiation. Do you agree? It's a form of energy. IR radiation, similar to light, the visible light. That's something called IR radiation. Visible light is having wavelength up to 800 nanometer, and IR will have a little bit higher than that one. IR is also called as a heat radiation. Just remember that one, right? And molecules will have a property to absorb light, right? Molecules likes to absorb light. And some molecules absorb some types of light. Some absorbs UV, some absorbs visible, some absorbs IR, some absorbs some other types of light. It's a property of the molecule depending on their electronic configuration, right? Now, let's take a water molecule as an example, okay? And this molecule can absorb IR radiation, okay? And because it absorbs IR radiation, it can have a vibrational transitions. It can vibrate, right? What do you mean by vibrate? The OH bond can be stretched like this one. Just to give one example. And this is a bond angle here, can you see? And that bond can bend like this, like a scissor, or it can wag like this one by absorbing the IR radiations. And that is called the IR transition. It's very specific to particular molecule, right? Depending on the stretching, bending, wagging, scissoring, different vibrational transition, you get different type of IR spectra. Just remember that molecules absorbs IR radiation and they vibrate, and the vibration will give you IR spectra. Keep that in mind, those who do not know IR spectroscopy. All right. We have C double bond O and we have NH. Can you see here? So the peptide bond, especially in the backbone, the peptide bond that is over here. Can you see? Right? You have NH and C double bond O. Can you see? And C double bond O, the bond can be stretched. Okay? And the CN bond can be stretched. And NH bond can be wag like this one, bending, right? So there is a stretching, and bending vibration. And that vibration will give you specific IR spectra. Okay? If there is a change in the structure of the protein, the vibrational spectra will also change. And the change in the vibrational spectra is directly proportional to the change in the secondary structure of the protein, because we are particularly focusing on the peptide bonds here, right? That will change because when you break hydrogen bonds and something, their local environment changes, then there is a change and the spectra is changed, and that gives us a direct information saying, okay, we are breaking the secondary structure of the protein. Okay? Especially in proteins, we are talking about amide bond. Please note that there's a lot of other bonds in proteins. But the characteristic bond for a protein to do the secondary structure is called amide bond. This is amide bond. You also call it as peptide bond. They are one and the same. Okay. There you have two transitions. There's a one here, the other here, and the other here. And this stretching is called amide 1 transition. Amide 1. And these two, the bending and stretching, this one here is called amide 2. This one, I hope I have a spectra. Yes, can you see? These are the two spectra of protein, amide 1 region. Basically, the amide 1 region is this one. Can you see this part? Okay, amide 1 region is this part where the stretching is happening, and then there is an absorption happening at around this particular wavelength. Can you see? And if there's a change in the bending of NH and CN, then the amide 2, the spectral pattern will change, because this particular vibrational transition will absorb in this particular region. Please note that it's very unique for the protein. It looks like a simple two peaks. No, it's very unique for the protein because the shape of this peak and the position of this peak determine the secondary structure of the protein. Make sense? So the shape and the position, they are very, very important. Okay, just to give that example here, look into this one. I have IR spectra of two different proteins here. The red one is the protein number 1, and the blue one is protein number 2. Maybe the red one, the protein is unheated, the blue one is the same protein heated. Now if you merge them together after getting the IR spectra, we take the IR spectra using this equipment. Okay, this equipment is used to get the IR spectra. You get the spectra like this one. Now just by superimposing them, you get a lot of information. It looks very simple, but it's very actually complicated. It says, okay, there is a difference. Can you see here? Okay, and also can you see the shape difference here? There's also some other part here also changing, but they're not very related to proteins, but yes, there's a difference, right? So there is a difference in the shape. There's also difference in the position, the location. right? And the shape and the location of these particular the peaks will give us a lot of information about the secondary structural change in a given protein. All right. So now I just to give more a little bit more information about secondary structure is made up of alpha helix, right, where you have a amino acids are coiled because of the intramolecular hydrogen bonding between C double bond O and NH of different amino acids. They pull together, that will help us to coil the proteins like this one, right? And they give the characteristic absorbance over here, 1652. Okay. Then if you have a beta pleated sheets in proteins, right? And they have a characteristics over here. Can you see? Then there's something also called random calls. Basically, they are always random. Difficult to say what is their folding and something weird ones. And they show characteristic IR peak over here, right? And these three information will be taken in the IR spectra, right? In addition to the shape. And that will be deconvoluted, and that will give us to the information about what is the percentage of alpha helix do you have, what is the percentage of beta sheets we have, what is the percentage of random calls we have. The change in the percentage of these things will talk about change in the secondary structure. Okay, please note that percentage calculation is not done often manually, it's always done automatically. There are some softwares, they'll quickly deconvolute these peaks and peak shapes, and that will tell you, okay, now you have 25% of beta sheet, now you have 60% of alpha helixes, you have only 0% of random coil. Okay, you can see that there are three proteins here, there is a difference between their beta sheets and alpha helix, and that will give us information about the secondary structure. Please note that this is not done manually, it's all automated today. You can perhaps do it. I never did it myself, but there are dedicated software to do this deconvolution. All right, let's move a little bit further. One of the interesting thing is that IR not necessarily can be used directly to determine the direct structure of the protein or the secondary structure. Rather, in food science, it can be used to find the difference, right? Untreated and treated. Polyphenol added, polyphenol not added, heated, not heated, ultrasound treated, not ultrasound not treated. Those differences can be seen very nicely by IR, right? Maybe it's very difficult to say the real secondary structure using IR unless you are a good person with all the software skills, but a normal person can actually see the difference between the two treatments using IR. What happens if I pasteurize my protein? Right? What happens if I do UHT treatment of my protein? Those small differences can be easily seen in IR spectroscopy because the heat is directly proportional to change in the protein structure. Change in the protein structure is directly proportional to the change in the secondary structure, which is directly proportional to the change in the IR amide 1 and amide 2 band. That is something I have shown you here. IR is very sensitive to temperature. Does that make sense? So I recommend you to use IR if you really want to see the difference between two samples in food science. Okay. One of the biggest disadvantages of IR for protein analysis is, especially for food samples, is water. Okay, that I have to highlight this particular point. For normal other people's sample, it's not a big problem because let's say organic people, they extract things in hexane or toluene or something like that, and they can dry it and they can completely remove water and they can dissolve it in another organic solvent which is not IR active, and then they can measure an IR spectra. For food scientists, is always a problem. Why? Because almost all of our samples contains water. Do you agree? There is no food samples contain. Even if you freeze dry, as soon as you remove from the bottle and you open, it absorbs moisture from the air. It becomes wet. So it's very difficult to find 0% water containing food samples. Almost impossible. Proteins are always high. Most of the proteins are hygroscopic. They absorb water molecule here and there as soon as you open the cap. And that gives you a biggest challenge. Why? Because the water has a particular absorption in this region, and this region is also the region for the amide 1 and amide 2. That's where the protein absorbs. Now that can superimpose your data, and that might mess up your data analysis. That is one of the biggest challenges. There are a lot of advantages here. What is the advantage according to you using IR? It's chemical free. Basically, that's one of the important thing is that IR is one of the easiest technique, right? You have your sample. There is no sample treatment except drying, right? You can also use liquid sample if there is no water, basically. I mean, you can use another liquid, D2O and other things, and you put into IR and press the button. Within a couple of minutes, I mean, even seconds, you will get the IR speed. The scan speed is so fast. Within a couple of minutes, you will get the IR spectra. No sample preparation, no addition of solvent, no filtration, nothing is needed. Put it, you get the data. That's the beauty. You require in the milligrams, exactly. Those you don't need something a very high concentrated. But for the food scientists, especially for the protein scientists, samples is not a problem. We always have a lot of proteins in our hand, right? So getting IR is very easy for us. Actually, that's true. But that's also one of the limitations I want to mention. IR cannot work if the concentration is too low. You get a very weak signal because IR intensity is also concentration dependent. If you say a concentration, if you have, let's say 0.001% protein, you get a peak like this one. 0.1%, you get the same thing like this one. And then the peak intensity changes depending on the concentration. And some people use this also for the quantification of the protein. I don't believe it strongly because it has its own problem. People claim it can be used for the quantification because IR intensity is directly proportional to the concentration of the protein. But there's a lot of problems. But just to let you know that if you have too little concentration, you don't get a good IR spectra. That's another problem. Maybe the water will overshadow the peak of your protein or your analyte of interest. All right, so I just give you some examples of advantages. Another advantage is IR equipment is always available in most of the laboratories. Not necessarily in the food science. You collaborate with your chemistry department, you get an IR instrument. So, and there's no much capital investment, and you don't pay for the samples also there because you don't use anything there, just except that sample chamber. But everything is free. Not free, but means you don't have to buy solvent and other things. But it's very easy. And that's another biggest advantage. This is an example showing that how different soy protein isolate, and when it's combined with the sugar units, glucosamines, and when it is reacted with an enzyme called transglutaminase. I'll explain transglutaminase in one of my lectures in the future. It's an enzyme. It will cross-link lysine and glutamine residues to make protein-protein cross-links, and they're trying to investigate exactly how that is changing the secondary structure. Just to give an example, how you can use IR to see the structural differences. As I said, food is never alone in food system. Protein is never alone in the food system. It is always interacting with something, and those interactions can be visualized using IR spectra. All right, now there is one more example here. What they were trying to do here is that they had this pea protein isolate and they used this glutaminase enzyme. You know that an enzyme called glutaminase. So you have a protein containing glutamine residues, which is having a structure like this one, glutamine, right? And which is having a positive charge. Now, if we use an glutaminase enzyme, and then it becomes glutamic acid. The glutamic acid is having COO minus. So basically, you can change the surface charge of the protein, right? And this might have a different solubility and different pI, and this might have a different solubility and different pI, and you basically change the functionality of the protein, in other words. And they were trying to understand, okay, when we change glutamine into glutamic acid, what is the secondary structure change? So they could evaluate that in this particular region. You can see that there is some change. I don't go deep into what exactly they have seen here, but so just to let you know that if you modify the protein using different chemical for enzymes, the secondary structure will change. And the change in the secondary structure can be evaluated with IR. All right, now we move on to the tertiary structure determination. Remember, the tertiary structure is nothing but it's a three-dimensional folding, and you have a disulfide bond, we have a hydrogen bonding, we have a hydrophobic interaction. Everything together makes protein to bend, and it will give you the tertiary structure. All right, so now there's a lot of different techniques to understand the tertiary structure. One of the easiest and the most common technique used today is fluorescence spectroscopy. Okay, it's cheap, rapid, easy. It has its own problems. I'll also tell you that one later, but it's an easy one. How many have studied fluorescence spectroscopy? No. Then again, I have to teach you the basics now. This is the proteins, right? Containing amino acids, right? And in IR spectroscopy, we mentioned about some of the bonds absorbs IR radiation and they start vibrating, right? And there are some molecules, they absorb UV light or visible light and they can excite their electrons to higher energy state. And then they will give you fluorescence, okay? There are 3 amino acids on protein, they absorb UV light very nicely. What are those? Tryptophan, tyrosine, and phenylalanine. They absorb UV light. Why? Because they have aromatic ring. Can you see? An indole ring, a phenyl ring, a benzene ring here, right? Because of these aromatic rings, they absorb ultraviolet light. You know what is UV light, right? What is the wavelength? 200 to 400, that is a nanometer. And usually the molecules absorb a particular wavelength or the range of wavelength. For example, the tryptophan has an UV spectra something like this, okay? And the maximum absorbance is around 280 and to 295 nanometer, okay? So they absorb a particular wavelength in the UV region, right? And when they absorb light, they go to higher energy state. I'll come back to that point. Remember that point. And tyrosine can also absorb light, the UV region, and also phenylalanine. But when compared to these 3 amino acids, tryptophan absorbs light in a very high efficiency. That's the right word. The quantum yield for absorption of UV light by tryptophan is very high. So we use tryptophan residue on a protein. This is tryptophan, for example, to understand the protein's tertiary structure change. Is it clear? Just to simplify things further, proteins contains a lot of amino acids. Among that, there's one amino acid called tryptophan. And tryptophan absorbs light in the UV region. Once it absorbs the light in the UV region, it can be excited and gives fluorescence. I'll get back to how it gives fluorescence later. And that property is used to understand the tertiary structure. How? Now, okay, I already have this one. Can you see the this one? Different absorption spectra for this one. This is a tryptophan, the red one, okay? And it absorbs around 295 nanometer in this case, all right? Now, I'll show you a little bit about, I'll show you something about how we can use this for the structure determination. Now, this is a 3-dimensionally folded protein, just you imagine. This is not chapati or dosa or something. This is 3-dimensionally folded proteins, okay? Right? And tryptophan can be present inside the protein. That means it's called buried inside. Let's take an example here. This is tryptophan amino acid, okay? This is protein. It can be present inside the protein like this one. What we call it as hydrophobic region. Because when you put this entire protein into water, the water is here, right? It's a hydrophilic region or aqueous region. The tryptophan can be present inside the protein. Now, it's not accessible to water. Can you see? Because water is outside, tryptophan is inside. So now it is in the hydrophobic region. Now, I'm heating this protein. I'm boiling the water. What happens? Bonds gets broken. Hydrogen gas bonds get broken. Protein will start opening up like this one. Now, can you see whom can you see now? Tryptophan. What do we have around here? Water molecule. Now, tryptophan starts interacting with the water molecule. Now, tryptophan fluorescence properties changes depending on whether it is in this form or whether it's in this form, right? Because the interaction with the surface, the microenvironment changes the fluorescence properties of tryptophan, and that is used to understand the tertiary structure of given protein. Basically, change in the tertiary structure. Please note, it's very difficult to say what is the tertiary structure. It is basically we determine the change in the tertiary structure. Is it clear? So, tryptophan, if it is buried, it's in hydrophobic region. It has a different fluorescence property. If it is opened up, it has a different fluorescence property, and that is measured in the fluorescence spectroscopy. Keep that in mind. Now, I'm going to teach you what do you mean by fluorescence. You know that nucleus in an atom, in a molecule, right? And we have somebody revolving around. Who is that? crazy guy, electron, right? So these electrons are just circling around the nucleus in three-dimensional orbital. There is all orbital theories you studied in your high school. Forget about it for now. But I can mention these electronic levels like this. Okay. Now I have a tryptophan. Right? Imagine tryptophan in its outermost orbital. It has 2 electrons. Electrons are shown as half arrows. Like, can you see? Now I take a UV light. I can take a UV torch, and it has a wavelength around 280 nanometer. Imagine. Okay. I shine this light on the UV on the tryptophan molecule in solution or in solid state, whatever, in solution, right? And then the light falls on the tryptophan now, right? When there is a sufficient amount of energy, the energy is in the form of light here, the UV light. Electrons absorbs the light, right? Once they absorb the light, what they do? They excite. They go to higher energy state because they have extra energy. They don't want to stay in the home. They just want to get out. That's what we do, right? I'm more energetic. I want to go to my friend's home and do a party, right? So I take that will excite to higher energy state, right? Now the electron came all the way here, right? Then basically when you go to a friend's home, you enjoy there, but you don't sleep there, right? Do you? Very rare, huh? You come back home, right? And that is called coming back to the ground state. But often you don't come back home directly. You go to some supermarket and have friends, another chat. So basically they come back to the another energy level like this by losing the energy in the form of heat. There's a lot of theory here. Because I don't have time, I will only show you the basics. Please understand that. So they lose the energy in the form of heat. Okay. And then they come back to the ground state. While they come back to the ground state, they release the energy back in the form of light again. Okay. And that is called fluorescence. I'm super bad with my spelling. Don't believe in my spelling, but understand what I say. Okay. So fluorescence. Is it clear? And that you can get as a spectra. You have an absorption spectra and you have an emission spectra, right? This is where the electrons are absorbing. You get a spectra. These are electrons are emitting back the energy, that you get a spectra. So it's called absorption spectra and you get a emission spectra. The emission spectra is basically a fluorescence emission spectra, and that depends upon where exactly tryptophan is present. I'll show that in later case. Is that clear? Now, I repeat this one again, is an important concept. So, electrons are usually in the ground state. By absorbing the light energy, UV light for tryptophan, it goes to higher energy state. Once they go to higher energy state, they not necessarily come back to the ground state directly. They lose the energy in the form of heat. That's called vibrational relaxation or in chemistry, but I don't teach you that one. A small amount of energy is lost as in the form of heat. And then they jump back to the ground state by losing the energy back, and that's called fluorescence. Okay, it's actually a unique property of a molecule. Every molecule absorbs a particular wavelength and emit at a particular wavelength if they are fluorescence active. That's a very unique. Tryptophan absorbs at 280, emits at 340, for example. If there is a change in the microenvironment of the tryptophan, then the absorption intensity or emission intensity and the wavelength will change, and that we will use for the fluorescence spectroscopy. Now, just to give my explanation here, this is an absorption spectra of tryptophan. Can you see? Okay, and this is an emission spectra of same tryptophan. This is absorption, this is emission spectra. Now, absorption spectra, the maxima is around, let's say, 295 nanometer. The emission spectra maxima around 345 nanometer. There is actually a difference. Can you see? Some portion of the energy is lost in the form of heat. Energy is lost in the form of heat. You know that energy is inversely proportional to wavelength. So basically, you lose the energy, so the wavelength is increasing, right? Because you lost some part of energy here, you absorb this much, right? And you lost this much as a heat, and the remaining is emitted, right? And that's the reason why the fluorescence spectra always having the higher wavelength region than the absorption spectra. All right, so now when the tryptophan is in the buried region and when the tryptophan is in the exposed region, they have a different absorption spectra. Can you see here? The absorption spectra of tryptophan varies depending on their microenvironment, how they are sitting on a protein, right? I have some small explanation here. Now, if the tryptophan is buried in the hydrophobic region, okay, basically they will show blue shift. What do you mean by blue shift? The emission maxima is toward the left hand side. Imagine the original emission maxima is, let's say, 345 nanometer, okay. If the tryptophan is more and more buried now, then the emission maxima will go in the left hand side, right? Now the same tryptophan is now under open condition like this one, exposed condition. Then it will have red shift. That means the emission maxima will go in this direction. And the change in the emission maxima, or the intensity, and also the position of the spectra will talk about the folding of the protein, or in other words, three-dimensional structure of the protein. In other words, denaturation of proteins. Have you heard about denaturation? Proteins will be denatured when you have when you do the heat treatment. Protein will be denatured when you do STS treatment. Protein will be denatured when you treat under urea. All this thing can be investigated using this technique, right? Because of the tryptophan's change in the emission properties. Okay, I told the right thing, right? Aqueous, you will have a red shift. When it's buried, hydrophobic region, you will have a blue shift. Just to summarize it again. So now basically imagine the original, this is your protein, untreated protein, for example, and you take a fluorescence spectra of this untreated protein. You ignite it at 295 nanometer and you measure the emission spectra. The emission spectra will look like this one, right? This is the emission spectra. Imagine the maxima is like 345 nanometer. Okay. Now, you did the heat treatment of this particular protein, right? And then it will open up, right? Now, the tryptophan is exposed. Now, it's interacting with the water molecule, right? Change in the microenvironment of the tryptophan will change the emission intensities and also emission, the type it emits, right? Then basically, if it's opened up, the emission maxima will come like this. So, that is called red shift. I think it's in chemistry you studied about bathochromic shift, hypsochromic shift, otherwise called red and blue shift, right? So this is called red shift, where you get a higher wavelength emission. So it may be like 365 nanometer now, just to give an example. Does that make sense? Now, for a reason, you did some experiment. Now the tryptophan is more buried, right? Compared to the previous one, it's completely buried now. Then you will get the maxima over here. It comes to 302 nanometer, imagine. And that information will tell us about the protein three-dimensional structure, denaturation, also renaturation. All right, so now these guys were trying to do the P protein isolate, and what they were trying to do is they're trying to investigate the ultrasound treatment. Remember the ultrasound assisted extraction? I mentioned it's a gentle technique, but there is a heat is involved, right? And some people also say that ultrasound generates hydroxyl radical. They're oxidizing in nature. They can oxidize the proteins. When the proteins are oxidized, the structure is changed, and that can be evaluated here. And these guys are trying to do that one. You can clearly see that imagine this is increasing the ultrasound treatment intensity. It's basically increasing amplitude and time here. They can clearly show you that, okay, you can see two things here in the spectra. Change in the intensity of the peak, and the second one is shift in the lambda max. Please note that these are two things always you look at the fluorescence spectroscopy. Usually the area does tell something about that one. If you know the deconvolution technique, is basically the intensity of the peak, which is again directly proportional to the area, and the shift in the lambda max, that talks about the protein conformational changes. As I mentioned, if the protein is more and more exposed to hydrophilic region, aqueous change, you will have the red shift. If the protein is more and more exposed to hydrophobic region, you will see the blue shift, right? And this paper, and some people also claim that if the tryptophan is in the hydrophobic region, you will have increased intensity. If tryptophan is more in the hydrophilic region, you will have a decreased intensity of emission intensity. Okay, but I tell you that don't believe much in the intensity, but always try to look into the lambda max, and that gives you the more clear information, especially when you have impure protein samples. In food science, it's a problem. right? When you have an ultra pure protein, you can talk about both intensity and emission maxima. But if you have an impure proper, it's very important to look into the change in the lambda max, because the change in the intensity is also depending on many other factors in fluorescence spectroscopy, especially the interferences. Okay. And this is another example where they're trying to investigate the protein polyphenol interaction. You remember my last slide, somebody was talking about the color and so on, right? Plant proteins having the biggest problem is the color. They're always colored, right? And most of us believe that this color is because of polyphenols or anthocyanins or tannins, right? And these guys were trying to investigate. What they did was they incubated pea protein isolate with quercetin. Quercetin is a phenolic compound, all right? And they saw, okay, increasing concentration of quercetin, they saw decreased fluorescence intensity, and they also saw change in the lambda max. Can you see? And that indicates them that, okay, now how quercetin is interacting with protein, and also whether the protein three-dimensional structure is changing because of this interaction, and that knowledge can be brought by this one. Okay. Before going to the next slide, and can you tell me a little bit about the advantages and disadvantages? I taught you a lot about fluorescence spectroscopy. All the amino acids will not absorb the light in that particular region, and all the amino acids are not necessarily fluorescent. It's only 3 amino acids are fluorescent. The fluorescence have a biggest disadvantage. Just look into this figure. Interaction with the other molecule. Usually, if someone else is absorbing at the same wavelength, then you will have a problem. You have a competitor in the solution, right? It's always a case in food science, right? Food is never a single molecule, I'm telling. You have protein with tryptophan, and you also have a polyphenols, imagine, right? Tryptophan is absorbing a light. Absorption is not a big problem because you can always put a lot of intensity, no problem. But once its emission happens, the emitted light can be absorbed by a polyphenol, which we call inner filter effect, right? And then everything will change. What you say and what you see and what is actually happening is different, right? The fluorescence spectroscopy data is a big mess in the literature. I'm sorry to tell this one. Okay, fluorescence spectroscopy in food science, I'm not telling in other part of the world, food science is a big mess, because sometimes they ignore the presence of some other molecules in food system, and they don't say about what these molecules are doing in fluorescence spectrum. So I'm openly telling now that don't believe fluorescence spectra data if they do not clearly explain how exactly they were measured, how exactly they have deconvoluted the data. Then don't believe in that data, and don't use fluorescence if you don't know exactly what you want to do. If you have a complex samples with polyphenols or absorbing molecules, you have to be extremely careful if you use fluorescence. It's not just because I have a fluorescence equipment, I can easily measure emission excitation, I get a beautiful graph, I can publish it. That's wrong, right? You need to understand what exactly you're doing, especially if you have polyphenols or vitamins and other absorbing molecules, because polyphenols, especially flavonoids, they absorb around 330 nanometer. Fluorescence emission is also in this range. Can you see? You have a problem. So emitted light can be reabsorbed even by the same protein also sometime. So that will give you a big problem. Now we shall move on a little bit more sophisticated techniques, but not in detail, just to showcase what instrument we can use or what technique we can use to get the real tertiary structure of a protein. One of the most well-established structure method is X-ray crystallography, what we call XRD technique. You can also use NMR to find out the real tertiary structures, but the most common technique today is X-ray. This technique is useful if and only if you can crystallize proteins into individual crystals, which is one of the biggest challenges, because not all proteins are crystallizable. If you can crystallize, organic molecules can be crystallized. If you go to organic synthesis laboratory, they try to crystallize their synthesized drugs or whatever, because they can actually talk about how exactly the structure look like. It's a very complementary technique to NMR at the end of the day. But crystallizing proteins is a nightmare. It takes a lot of efforts. If you somehow can crystallize individual proteins, then you will get a fantastic 3D structure of a protein, and that is something people use to develop the databases. You must be knowing that the protein data bank, PDBs and so on, where you get a lot of information about amino acid sequence and the structures of a known and unknown proteins and so on, they all often come from this kind of methods, that they do the crystallization, they get the structural information, they get the sequence information, then they understand the folding and so on. If you recall, there was a Nobel Prize last year on protein structural elucidation, if you remember. You can also use de novo sequencing and modern sophisticated artificial intelligence technique to find out the three-dimensional and also quaternary structure of proteins. XRD is a not simple technique, it's a complicated technique. You get a protein and you just crystallize them, and then you get the X-ray diffraction data using an XRD equipment, a very expensive unit. I don't know, we have it in every institution here in India, but it's also a specialized instrument, is only found in specialized laboratories. And based on that, you can get the electron density. Basically, you know that the protein is folded like this, right? Every atom or every molecule, every atom, in this case amino acids, they have a special electron density. It's a unique for that particular atom. It's unique for that particular amino acid, and that density information can be captured using sophisticated software, and that will help us to develop the protein three-dimensional structure. I will not go any detail into this technique because there is a professor coming in the coming days who will give you an exclusive lecture on X-ray crystallography, how we can use XRD to determine protein structure. So it's not my job now, but I'm just telling that we can do these things using XRD. Is it okay? So this is how the X-ray crystal structure look like on a computer, right? After deconvoluting everything, so these ribbons and all these things, you can see beta sheets and random coils, and so all this information often comes from XRD, the diffraction factor pattern. This is one of the nicest structures showing the quaternary structure of soybean corn glycinin. So you can, I think it's a beta heterotrimer, homotrimer. So you can see the individual protein subunits. Can you see? That is showcased as a different color. It's just an interpretation. Please note that protein does not have a real color. It's just to showcase you in the computer, on a computer screen, we show it in this way. Okay. And if you go to the PDB or some kind of a software, you can actually turn and tilt and see how exactly the protein looked like on a three-dimensional level. I can't show it on my computer, unfortunately, on a PowerPoint, but you can actually tilt and see how exactly the protein looked like on a four-dimensional way, right? And where is the space inside, where is the random coil floating inside. You can also pinpoint where exactly that tryptophan, the individual tryptophan, is located. Oh, now it's buried or it's on the surface using this X-ray crystallographic technique. It's another structure of moringa. I just wanted to show that the protein can be as big as this in corn glycinin, very complicated. It could be also very simple protein. But to get this structure, crystallization is very, very important. It's a bottleneck often. I'm going to teach you one of the most interesting and important technique, that is STS page. A very simple technique, but gives you one of the most, it gives you the overwhelming amount of information about proteins. Doesn't matter whether it's milk protein, plant protein, animal protein, bacterial protein, fungal protein, you can use STS page irrespective of protein type. Okay, so in the previous slides, we are talking mostly about getting the structural information, but STS will give you more about the compositional information. It doesn't talk a lot about structure. It does in certain cases, but usually it talks a lot about protein composition, means what type of proteins you have. For example, in my very first class, I mentioned to you about soy contains globulins, right? Corn glycinin and glycinin. Rapeseed contains nepin and cruciferin, and our pea protein contains some other type of protein, including prolamines and so on. And how do we visualize them? How do we separate that different subclasses of proteins? For that, you can use STS page. Well, what is STS? STS is sodium dodecyl sulfate, which is an anionic detergent. Okay, STS is an anion. Anion is negatively charged, right? So it's an anion detergent. You all know that the detergent, what does they do? They clean things, right? Yeah. So basically, this detergent will clean proteins. In other words, it will denature proteins. Okay. It disturbs the hydrogen bonding or hydrophobic interactions on the protein. The protein is three-dimensionally folded like this one, right? Now, if you add an STS to this one, the protein will become like this. Okay. They don't cleave the disulfide bond. Remember, STS cannot cleave the disulfide bond. It can only cleave the weaker interactions. All right. And that will denature the protein. That's the first step you have to keep in mind. Okay. What exactly we do here is that now you have a protein sample. This is our protein sample in a tube. Okay. And we add STS. Okay. And we also heat sometime around 90 degrees Celsius for 10 minutes, depending on the situation. And then the protein will be denatured like this, right? Not only the protein will be denatured, since it is an anionic detergent, it absorbs on the surface of the proteins and it will give the net negative charge on the protein surface. Now the protein will be negatively charged. Am I clear? STS will do two things. The first one, it will denature the protein. And the second one, it will give you the surface negative charge on the protein. Protein can have positive, negative, everything depending on the pI and the pH of the solution. But as soon as you add into STS, it will get mostly negative charge. Keep that in mind. All right. Now, I have a protein solution containing negatively charged proteins. All right. And now I take that proteins, I just want to have some space, put it in a gel. This is a gel, okay? And the gel is actually made up of polyacrylamide, which is a polymer polysaccharide, polyacrylamide gel, okay? And the gel contains small wells like this one. Okay, you can have a number of wells depending on the gel you prepare. You can prepare this gel in the lab, your own, or you can buy from from the supermarket, but from the chemical company. You can buy them. Okay. So now I will load the sample to one of the wells here. Okay. A small amount. We're talking about 6 to 10 micron liters containing the protein amount of some micrograms only. It's not a high amount you're talking about. And the entire gel, I'm going to place in a beaker or a chamber containing some kind of buffer. Okay, entire thing will be placed in a chamber containing buffer. Now, this chamber can be connected to some sort of electricity, right? It will be connected to positive electrode and also it can be connected to negative electrode, something like this. Okay, and that will go to an electrical system like this. Now, there's a switch here, right? I turn on the switch. What would happen? Okay, so as per the charge of the protein, it will separate. And which direction the protein should move now? Downwards, right? The protein will start moving in this direction. Why? Because remember, ST has given protein a negative charge and you have positive electrode in this one. We have studied in our physics that negative loves positive. So it just wants to go towards the cathode, right? Moves in this direction. So protein will separate based on the charge. But is it only the charge? Molecular weight or the size of the protein also does matter. Who will run faster? Those who are having the less molecular weight or the size will move faster. The bulkier stays behind. Okay. Now, here it's basically a mixture of protein containing small and bigger proteins and they are loading it as a mix here, right? Because of the size and the charge difference, the bigger one will stay here and the smaller one will come here. So basically, based on the size, protein will separate as a band. We call it as STS page protein bands, right? Does protein has a color? No. But when you do this one, can you see the bands with your naked eye? Exactly, because our eyes can only see apparently visible light, right? And the proteins does not absorb in the visible light, so you can't see proteins unless you're God, right? So what we have to do is that we need to make protein somehow colorful, and then after doing the electrophoresis, the whole technique is called electrophoresis, okay? We put that in a staining container containing some kind of a dye called Kumasi Brilliant Blue. I don't write the full name because I have no idea about spelling. Okay, Kumasi Brilliant Blue. That's a Kumasi staining. Is it CU something? Okay, I write some wrong spelling. I said you that I'm a bad with spelling. There's a Kumasi dye. So it's actually giving a blue. It's a blue dye actually, and it loves proteins and it attacks the protein. Then you get the band as a blue color band. It looks exactly like this one. What we do in real life is that in parallel to your sample, we run a molecular marker. The molecular markers are nothing but they are the proteins with known molecular weight. The company will tell you this band is equivalent to 14,000 Dalton and this band is equivalent to 2,000 kilodalton because the company has already measured it when you buy the band. So we run the molecular markers in parallel. The molecular markers is basically a mixture of different proteins with different molecular sizes. So that will also run along with your sample and that will help us to understand which protein, what is the molecular weight of my protein. Now you can see in this case my protein is this one. Imagine. I can tell that okay, my protein may be in this particular band, maybe between 31,000 to 45,000. I say maybe the molecular weight of my protein is 40,000. Do you agree? And that's how you use STS page to find out apparent molecular weight, not the real one, apparent one, approximate molecular weight you can actually determine. I use this word approximate. Don't tell anybody that STS can be used to find out the right molecular weight. No, it just tell you an approximate molecular weight. But that information is very important for us because we talk about big proteins and small proteins in food science. So that can be easily visualized by STS page. Now, I'm just giving some example how we are using in alternative protein world STS page. This is a nice STS page I found from this particular article. You can see that how we can separate different soy protein subunits. For example, corn glycinin having subunits can be separated. The subunits are separated because when you have because soy protein has let's say corn glycinin, let's say it has two subunits, it has multiple subunits. Because of STS you add, the interaction between them is broken and the subunit becomes individual subunit and they migrate differently. Can you see here? So now STS can also be used to understand what kind of subunits a given protein has, right? And it also helps us to understand different kind of proteins. And you can also use STS for the more complicated stuff. This guy tried to separate all these allergens. Allergens are proteins. I'll talk about that later in other class. So you can also separate allergens and see what kind of allergens you have. But please note, one of the important thing is that by looking into the band, you don't get the information. Because the only information I have the marker here. It's all about finding back to the literature, right? That's very important in STS page. So you just see a bands and you just see, okay, molecular weight. What are those? Nobody knows that idea. You just have to go back and see the literature. Who is telling, okay, this person is also getting the same band. Usually, a 99% chance that the STS page look alike, even if you do in this lab and the other lab, provided you use the same condition. The bands will not change their order. The order is always same. The marker will always move the same way. It's usually a very robust technique. So that will help us to understand, okay, this band belongs to corn glycinin, this band belongs to glycinin, this is a beta lactoglobulin, this is alpha lactalbumin. That information can be bought. So not directly, but indirectly. Is it okay? Clear? So this is a pea protein isolate. We can see that how they are separating corn albumins and corn vicilins and other proteins. And this is a nice STS page gel picture of a rapeseed protein isolate. It contains cruciferin subunits and nepin, a smaller protein. Cruciferin is around, I think, approximately 300,000 Dalton, and nepin is around 14,000 Dalton. So basically, that can be separated. Okay, and you can also use STS page to see the impact of protein modification. For example, you're doing ultrasound treatment. Then because you may break the proteins and that can be visualized. And you are using extraction. For example, we talked about salt extraction. We talked about alkali extraction, right, in yesterday's class. Now in the salt extraction and alkali, this is salt extraction and alkali extraction. Maybe the protein extraction yield is the same or different. Not only the yield, the type of protein you extract is different usually. Maybe salt is selectively extracting salt-soluble proteins, and alkali extraction does not care, it extracts everything. You want to see what is the difference between the composition. It's not always about the yield when it comes to food science. It's also about the composition. So the composition variation, depending on the type of extraction, that you can use STS page. If you're doing some extraction studies, I really recommend. It's a nice add-on to your manuscript, basically, just to see the quality of the protein you're extracting. Another important advantage of STS page is we can also determine whether there is a disulfide link in the protein. Here is an example of a disulfide bond. This is you can see the intramolecular disulfide bond here. I really want to show whether there is an intramolecular disulfide bond. How do I show that? Please note that these disulfide bonds are reducible. Okay, that means this is how the disulfide bond actually exists on a protein, right? If you use a reducing agent, it will break this disulfide bond and it will give you 2-thiol group like this one. Basically, the protein was like this and it becomes like this, right? Originally, it was 100 kilodalton. Imagine this subunit is 25 kilodalton and this subunit is, let's say, 75 kilodalton. You get two units. One is 75, other one is 25, right? What do we do in reality is that we run STS page twice or in two different wells, right? One we will run without adding a reducing agent, negative, no reducing agent. The other one is with reducing agent treated sample. Without adding reducing agent, the sample will give a band here. If you treated the sample earlier with the reducing agent, where do you get? At the lower band, right? Yeah, one you will get at 25 and one you will get 75. You see, get nothing here, right? And this information is extremely crucial for protein scientists. That will tell us whether your protein is having disulfide cross-linking, because disulfide cross-linking is an important cross-linking that happens in food system to create aggregates, what she was telling. Protein can make a bigger, larger aggregate with the better functional properties depending on the disulfide linking. And that disulfide linking we can visualize nicely with this particular method. Is it clear? We have to run samples 2 times. One, first you do not reduce, just run directly. Second one, use a reducing agent. For example, DTT, dithiotriol. We use that one, or mercaptoethanol. We can use these two reagents to reduce the protein and then run the reduced sample. If you see the band disappearance and formation of new band, which clearly indicates that you have a disulfide linkages in your protein. Okay, this is an example to show you here. They are running 2 gels here under reducing condition and non-reducing condition. You can see that they get high molecular weight stuff in the non-reducing condition, and this is broken into different pieces. Then you can clearly say that these proteins actually containing disulfide bonds, because you get a lower molecular weight fragments. Is it clear? So just to give an example here, this is again the soy protein isolate. If you run it in the non-reducing condition and this is reducing condition, you get a different pattern. Basically, these higher molecular weight fractions, as soon as you add a reducing agent, they are broken and get the lower molecular weight fragments. It clearly shows that soy protein isolate has a lot of disulfide bonds. Okay, so that information you can get easily with STS page. So whenever you want to run STS page, always run in 2 ways, non-reducing and reducing. You simply get an extra information. All right, so remember this slide last time, protein polyphenol interaction. Polyphenols can interact with proteins and can damage the protein or improve the solubility, and those things can be visualized. I'm going to show you another technique now. This is a complementary to the STS page. Now we have a protein here, okay, and the polyphenol is attached to the protein, right? And remember, polyphenols will always have a functional group, OH. Now I'm talking about protein polyphenol covalent bonds. Please note, it's not a non-covalent interaction, it's a covalent interaction, right? Now I have a 2 protein, one attached with polyphenol, one without polyphenol. Can you see? This is with polyphenol and without polyphenol, right? I run STS page of them. I don't have a real experiment with the plant proteins, but this experiment we have done with the milk proteins. I'll show you how we can use this technique. This is my own result. We took the milk and we added green tea extract. You know what is green tea extract? Which contains a lot of polyphenols, epicatechin, epigallocatechin, and all these catechins you have it, right? And we also know that the milk proteins interact with the polyphenols nicely and make a covalent adduct. Okay, then we ran through the STS and you get a separation. Now this bands actually contains protein plus polyphenol together attached. Anything that is non-attached will run away because the separation takes place only for the proteins in STS page. All the free polyphenols which is not bonded will vanish, which will just come to the buffer here. Do you agree? Now the bands what you see, it contains protein plus polyphenols. Now if I take this one and put it into the Kumasi staining, what do I see? I see exactly the blue bands, right? But can I differentiate polyphenols there? I just see again the same protein band, but I really want to see whether protein is now attached to the or bonded to the polyphenols. How do we do that? This is a technique we use in our laboratory. We use a technique called Western blotting. It's a common technique in molecular biology. What do we do in the Western blotting? We take the STS gel and we put the nitrocellulose paper or what is it? We also use PVDF, right? Nitrocellulose or PVDF paper, and we transfer all the band to the paper now, right? That's actually blotting instrument is there. It will quantitatively transfer the bands into the paper now, right? Now we get a Western blotting. I hope I have a picture here. We get a paper like this. Now all the bands are on the paper, right? This is called nitrocellulose paper. Nitrocellulose paper, right? And now this paper, now quantitatively transferring all the bands here. Now this bands contains again protein attached to polyphenols. And then we put this paper, we spray a reagent called NBT. NBT is nitro blue tetrazoleum. Okay, nitro blue tetrazoleum, which is actually a redox reagent. It likes to interact with the polyphenols. Now the polyphenol attached on the protein contain hydroxylic group. It's an antioxidant. Polyphenols are antioxidant because it has OH group. Now this will interact with these polyphenols and it will be reduced to form a formazan, and that color is actually purple. Now if you see a purple band on the protein, it indicates that protein is attached to the polyphenol. And this is a fantastic, easy technique to use to understand protein polyphenol interaction. You're welcome to use it. Is it clear? It's a complementary technique to STS page. STS page will only give information about protein, nothing else. Now I want to see the protein polyphenol interaction. What do? I transfer all the protein bands into the nitrocellulose paper, and then I stain it with a reagent called NBT. NBT will be reduced to formazan. The formazan is actually having the color purple. And if I see the purple color band, which clearly indicates I have a protein polyphenol interaction. Is it clear? But one of the disadvantages is that if your protein is glycated, Maillard reaction, they also give a purple color. Be careful. Okay, I'm not telling it selective to polyphenol, right? That's one of the disadvantages here. Is it clear?